Hello everyone and welcome to today's Biology Daily Booster. This is number 13 in our Paper 2 series, and while it's not listed on the advanced information, that doesn't mean there won't be questions on it, as we know from Paper 1. What we're going to do then is have a look at genetic engineering to start. When we talk about genetic engineering, this is our process of altering an organism's genome by selecting genes that create a certain characteristic. So what we're doing is changing both the genotype and the phenotype. Some examples of where we've genetically engineered some organisms then. We've genetically engineered cotton to increase the yield of the crop. We've engineered our corn so that we can produce toxins to kill insects that would normally damage that crop. And in bacteria, we've actually genetically engineered them so they produce things like human insulin for us. The very basic overview of what we're doing here then is we take foreign genes from an organism that show the characteristic we want, and then we insert those into plant or animal cells at that early stage of development. They will then demonstrate that characteristic. There are some risks of this process. Firstly, we don't know the long-term effects because we've not had this for a long time. So we don't know if you're eating genetically engineered food for 50, 60 years of your life, what effect that could have, because it's not been around that long. There are some possible negative effects, obviously not proven yet. Firstly, health problems. We might be introducing new allergens into food. So if you've got an allergy to a particular thing that's not normally in tomatoes, but we then insert that gene into tomatoes, could cause some new allergies. And we could actually have cross-pollination with wild plants, which disrupts the balance of our ecosystem. If we look at how this occurs in a bit more detail then, so we're going to use the example of how we make human insulin using bacteria. What we're going to do then is we start off here with our human cell. What we're going to do is take the DNA and then we're going to cut the gene that makes human insulin from the DNA using what we refer to as restriction enzymes. So restriction enzymes cut the human insulin gene from the DNA. From a bacterial cell down here, we're going to remove the plasmid. So that loop of bacterial DNA, we remove the plasmid and we use the same restriction enzymes to cut that plasmid open. What we then do is we place our human insulin gene and our open plasmid together with an enzyme called DNA ligase, and it's going to join those two things together. We then introduce that plasmid back into our bacterial cell, and then we can grow them in large numbers. And remember, because bacteria produce genetically identical offspring, every bacterial cell that comes from that one will have the human insulin gene as well. Key things we need to remember, folks. Restriction enzymes are the ones that cut the DNA, and they're going to leave these things called sticky ends. Now, a sticky end, quite simply, is just where we've got a little sequence that has a little bit sticking out. So you can see this little bit here, we've taken those bases off so that when we've used that same restriction enzyme to cut the plasmid, they're going to be complementary, because on our plasmid we're going to have these ones here. We'll have a G and a C, and then the rest of the molecule, just put in some random bases, and that means they're going to be complementary bases to each other. So that's how they then join through the DNA ligase. So ligase joins, restriction enzymes cut. It's all good and fine saying we're going to do this, but we can't just assume that it's taken up the gene we wanted it to, because it's not a perfect process. We need to check. There's two ways that we can check, but they both use what we refer to as a marker gene. So the two options for marker genes are antibiotic resistance or fluorescence. If we look at how we use the antibiotic resistance gene first of all, what we do is at the same time that we're inserting our human insulin gene, for example, we also insert a gene for antibiotic resistance. That means that if it takes up the insulin gene, it's taking up the antibiotic resistance gene as well, okay? We then place that bacteria onto an agar plate which contains the antibiotic it's meant to be resistant to, incubate it, give it time to grow, and then when you take out that agar plate, any colonies present will have the antibiotic resistance gene. So they will have the human insulin gene too. If they don't exist on the plate, it's because they weren't resistant to that antibiotic and therefore they didn't have the human insulin gene anyway. Second option, the fluorescent marker gene. Again, we insert our marker at the same time as the gene for our desired characteristic, transfer the bacteria to an agar plate, incubate it, time to grow. This time though, what we do is we shine a UV light onto it, and any of those colonies that glow have the genes. If they don't glow, they don't have them. So all we then do is select those colonies with the glowing, and then we grow those in large numbers. We're then going to have a little look at the idea of biotechnology. When we talk about biotechnology, this is using biological processes or living organisms to manufacture products we want. So a great example here are genetically modified organisms. There are two very successful genetically modified crops that we use today. First one, golden rice. We've taken a gene from a daffodil and placed it into a rice plant. Why, you might think? Well, because that rice then produces the chemical beta-carotene. Now, beta-carotene is necessary for the body to make vitamin A, which you need for good eyesight. We will then be able to grow this golden rice in areas where they don't necessarily have good access to a range of vegetables that would have beta-carotene present. So those areas of the world have had massive problems with blindness in particularly children, because they just didn't get the beta-carotene, couldn't make the vitamin A, went blind as a result. So by swapping out just standard rice for golden rice, we can actually solve that bit of the problem. Second one, BT corn. This is where we take a gene from a bacterium and we're going to put it into maize. Now, maize is just a crop plant that we use, a cereal plant. What that does is the maize then produces a protein which is poisonous to insects. As a result, the insects won't damage our maize plants. We get a much higher yield from that field. One phrase that we should know is the word vector. A vector is something that transfers foreign DNA to an organism, and there's a range of different vectors we can use. Bacteria, the virus, a plasmid, those are all vectors because they're transferring foreign DNA to another organism. And you can see the little example down here. So we've got our restriction enzyme cutting out the gene we want. We've got our vector here that we're going to also use the same restriction enzyme on. We insert the actual gene into our vector's DNA, and then that vector introduces it into a plant cell through the process of insertion. So that's just a little kind of virus, if you like, down here. So all we've done there, introduced our actual DNA we want into the virus. The virus then inserts the DNA into the actual plant cell, and the plant then is resistant to that particular issue. As always, if there are bits you want to go over in more detail, you can obviously use the playlist on the main channel to help you. Use your revision guide, your notes. Do have a look at some of the past exam papers just to get a bit of a feel for the kind of questions that come up, and use the mark scheme so you can see what the marks are awarded for. And of course, don't forget to join us again tomorrow for our next Daily Booster.